All right. Give me 1 second, actually. I have to turn on my light. Make me look pretty. All right, there we are. So, BNGO is on another huge bull rally this morning, just like we saw yesterday. And if we back up to the 2-day 5-minute chart, you'll see right now it's very bullish. And pretty much every time it goes into oversold territory, it bounces up. Same thing happened today. Same thing happened around this area. So overall, general sentiment seems to be extremely bullish right now. And there's a few catalysts, I think, behind this today. Of course, the last day of the symposium. Yeah, the potential of ARK investing into the stock. But in my opinion, at least for right now, at these prices, I'm not sure if ARK is going to buy. Supposedly, Simon from ARK G has a podcast that'll be coming out today, and we'll learn more about his new updated opinion on their technology. Hopefully. That's just what I've heard at least, and I could be wrong. Anyway, just to start this off, I just want to say that I'm not invested into the stock. I don't hold a position, long or short. Just trying to give you the best information possible, and I'm going to leave it up to the scientists to try to explain what exactly they're doing in their research and to help you make a decision on whether or not you should buy in. So, with that said, let me pull up the symposium and we'll get this thing started.
He will be giving the introduction to the technology and giving details about our session today. Thank you. Alex, all yours.
Okay, thank you so much, Elka. Today we have an exciting session, which is the COVID-19 host genome SV expert panel session in Q&A. At the beginning of 2020, 2020, the world experienced the beginning of a pandemic, the COVID-19 pandemic, and the scientific community really took action as quickly as they could to get involved and to help understand this new disease. This is showing work published by the scientific community over the year and related to COVID-19. The publications increased over the first few months and then really kind of plateaued at about 2,000 per week. So this is a tremendous amount of research involving COVID-19, and a lot of these studies address the relationship between the host genome and the virus. However, the impact of structural variation with the host genome interactions was absent. Going through a few of the studies quickly, a lot of these studies did focus on the host genome, on really the single nucleotide variant level. There first were several GWAS studies, this one finding associations with chromosome 3P and 9Q, for example. Other GWAS studies finding other linkage groups. There were studies, for example, this study from the Netherlands where whole exome sequencing was used to find variants that would impact the severity of symptoms, in this case finding a loss of function mutation in TLR7 in young men who had severe COVID. Additional genome-wide variation studies found other loss of function variants involving genes in different host genome candidate genes involving immunity. However, none of these studies address
Welcome everyone to day 5 of our next generation cytogenomics symposium. And today we have a very special session on COVID-19 host genome structural variation expert panel session. And I would like to introduce Dr. Alex Hasty, who is the Vice President of Clinical Affairs at Bionano Genomics. And today he will be giving the introduction to the technology and giving details about our session today. Thank you. Alex, all yours.
Okay, thank you so much, Elka. Today we have an exciting session, which is the COVID-19 host genome SV expert panel session in Q&A. At the beginning of 2020, the world experienced the beginning of a pandemic, the COVID-19 pandemic, and the scientific community really took action as quickly as they could to get involved and to help understand this new disease. This is showing work published by the scientific community over the year and related to COVID-19. The publications increased over the first few months and then really kind of plateaued at about 2,000 per week. So this is a tremendous amount of research involving COVID-19, and a lot of these studies address the relationship between the host genome and the virus. However, the impact of structural variation with the host genome interactions was absent. Going through a few of the studies quickly, a lot of these studies did focus on the host genome, on really the single nucleotide variant level. There first were several GWAS studies, this one finding associations with chromosome 3P and 9Q, for example. Other GWAS studies finding other linkage groups. There were studies, for example, this study from the Netherlands, where whole exome sequencing was used to find variants that would impact the severity of symptoms. In this case, finding a loss of function mutation in TLR7 in young men who had severe COVID. Additional genome-wide variation studies found other loss of function variants involving genes in different host genome candidate genes involving immunity. However, none of these studies addressed structural variation. And so in our discussions as a structural variation company, we we had a lot of discussions with other people interested in structural variation, and so we and some of our customers and our collaborators decided to set about on creating a host genome structural variation consortium, and some of the members of that consortium will be talking today and giving a lot more details about how structural variation could be involved in the host genome interaction with the SARS-CoV-2 virus. I'm going to present and provide a background on the Bionano Genomics technology to introduce our technology and remind people of how this technology platform works. So we are a structural variation company. We do not look at single nucleotide variants or polymorphisms. We are able to assay for structural variations from hundreds of base pairs up to millions of base pairs. And we can assay for structural variants of all different classes. This diagram shows the different structural variation classes, such as deletions, insertions, repeat expansions, inversions, and translocations, as well as aneuploidies. The basis of the Bionano technology is really shown in this video and this figure here. What the technology does is visualize very long DNA molecules that have been linearized in a nanochannel array. The video on the left is showing DNA molecules in green being loaded and linearized through basically like a comb that separates and allows the DNA to be fed into nanochannels as single molecules that are very straight. And once the nanochannels are filled, the electrophoresis would stop, and we would take pictures of those DNA molecules that are hundreds of kilobases to megabases in length. And the pictures look like this. The top electron micrograph is showing the actual nanochannels where the DNA is linearized. This is all done automatically in the Bionano Saphyr system. Showing the overall workflow quickly, the workflow starts with isolation of very long DNA molecules from cells or tissues. Then the application of a fluorophore, which is conjugated to a 6-base sequence motif, which is recognized by an enzyme, which in a and non-damaging way will transfer the fluorophore to the DNA molecule at each 6 base sequence motif. This occurs every 5 kilobases across the genome on average. And the pattern, the distances between those sequence motifs and the pattern that it makes creates a unique pattern. The DNA is loaded onto a Saphyr chip, which contains the nanochannel array, and loaded into the Saphyr system, where the DNA is electrophoresed into the nanochannels to linearize and then image the DNA. This is all an automated process. After imaging the DNA, the locations of the fluorophores and the DNA molecules are extracted from the image, and we can extract this unique pattern of labels. That pattern of labels can be assembled into a consensus map that gives a unique pattern for the given region of the genome, and then that map can be compared to a reference genome such as HG19 or HG38. After comparing a sample's map to the reference genome, we can automatically call structural variants based on differences between the map and the genome. For example, this represents an insertion. This could be 500 base pairs up to many kilobases or megabases. And the technology can recognize those insertions and measure those links. The technology is able to find all classes of structural variants. So this shows the different types of structural variants that can be found. In each of these figures, the Bionano map is in blue and the reference genome is in green. A deletion would look like this, where part of the reference is absent from the case's DNA, the sample's DNA. An insertion would be additional sequence in the sample compared to the reference. We can measure repeat expansions and contractions accurately, even though these are difficult to measure by molecular method. Since we have very long DNA molecules that span those repeat tracks, we can accurately measure the size of those expansions and contractions. We can find duplications as well as the position of the extra copy. So this represents a duplication of the segment in the reference here, where one copy is here, and the additional copy, the second copy, is adjacent that in the same orientation. So this represents a tandem direct duplication. If that had been inverted, we would recognize that as well. And if the duplication lands on another chromosome, we would also be able to recognize that. This technology can also find balanced structural variants, which are the most difficult to find in general. This shows a translocation where the sample's map aligns to 2 different chromosomes, and you can find the breakpoints on the 2 chromosomes and the position where it's fused based on the map pattern. Inversions as well can be easily detected by recognizing an inverted orientation in the sample compared to the reference. At the same time, we have a second algorithm run in parallel, which can find copy number variants based on the relative number of sequence motifs, labeled molecules that align across the genome, such that copy number increases and decreases could be detected automatically from the software. All of the structural variants that are found in a sample are displayed on a circus plot, as well as which is an interface to navigate to more detail on the structural variants. This is a genome-wide circus plot, which shows a 1122 translocation, for example. It also shows the copy number profile and the interstitial structural variants on that plot. So this is a convenient visualization system that allows us to navigate into each individual structural variant just by clicking on the points. With that, we will begin our session today. We have a great lineup of speakers that are experts in structural variation, as well as different types of sequencing, and have a wide array of experiences. We will be it started with Dr. Ravi Kole from Augusta University, who will give an overview of the consortium. This will be followed by Dr. Siavash Riasi from UC San Diego, who will be talking about fandom and an approach to validate Saphyr structural variations. Dr. Silviu Alan Beckenau from VCU will talk about GWAS studies on COVID-19 host genetics. Dr. Alex Hoyshan from Radboud UMC will be talking about rare genetic variants, which may predispose to severe COVID-19. Dr. Michael Zodi from the New York Genome Center will be speaking to us about whole genome sequence studies on COVID-19 host genetics. Dr. Eric Jarvis from Rockefeller University will be talking about comparative genomics in SARS-CoV susceptible patients and animals. Dr. Alan Beggs and Dr. Catherine Brownstein from Boston Children's Hospital will be speaking about structural variation analysis of children with MIS-C. And Dr. Ravindra Kole from Augusta will finish the session with results from the first Saphyr study on ICU-admitted patients. So with that, I will now be turning it over to Dr. Ravindra Kole for the first session. And if people are tweeting, please use the hashtag Bionano Symposium. Thank you very much for joining our session today.
Hello, everyone, and welcome to the Genomic Symposium. Today is a COVID-19 day, and in today's session, we will be discussing mostly our findings and approaches to understand the COVID-19 host genome. My name is Ravindra Kole. I'm a pathologist at Medical College of Georgia and Augusta University. And I'm going to introduce our group, our consortium, and introduce the speakers. As you know, COVID-19 pandemic has affected pretty much every part of the world. As I tell my kids, this probably is the biggest challenge we have faced since I was born. And we're doing pretty much each and every specialty in medicine is working towards understanding the disease and working towards getting the better outcome. As of now, more than 88 million people have been affected, and more than 1.9 million people have died. But one of the things which is fascinating about this disease is the split mortality. This is what I like to call you have this huge amount of individuals who are infected, but an extremely small percentage of them become symptomatic. And then in that symptomatic, really a small percentage end up dying. People have associated a lot of clinical comorbidities with the mortality. But again, even if you look at race, gender, age, or comorbidities, it's becoming challenging to explain all of these mortalities. We would have a younger population, less than 30 years old, without any comorbidities, dying in comparison to with the older population with the comorbidities. So as of now, majority of the meta-analysis have shown that the individuals who are hospitalized have 31% of diabetes, 50% of hypertension, and around 14% of heart failure. We know that the Black Americans are dying as twice the rate of the white Americans, and definitely the children, they tend to have very low negative infection rate, but they do have their own category of inflammatory syndrome, which we call as multisystem inflammatory syndrome in children. And there has been a lot of global efforts in understanding the host genomics. Why do some individuals are affected worsely in comparison to the majority? As I said, there are multiple global efforts, and one of the biggest one is the COVID-19 host genome and genetics initiative. And we are being following the data from there. And one of the challenges which we identified based on the information which is already there public out of this initiative is these genetic studies are based on GWAS or whole exome sequencing, and they cannot detect larger variants. And then on the other hand, some of these variants, when we looked at it, which were identified, they were mostly severely associated with immune deficiency or immune defect. So we acknowledge what was happening around us, and one of the idea behind what we're doing right now was to see whether we can add to the existing knowledge base in understanding the split survival for COVID-19 individuals. So we get together, a couple of friends together, we're working, and we identified that there is a definite need and a knowledge gap with understanding the structural variants in these individuals who are severely affected by COVID-19. And we ended up putting together a group of labs who had a very similar interest, and we created a consortium called COVID-19 Host Genome Structural Variant Consortium. The idea behind this consortium was to put together a group of people who are working in a structural variant lab, so understanding the structural variants both in vertebrates as well as humans, and then see what we can identify in these groups. So some of the objectives of our consortium are, number one, we want to create a global networks of labs who use novel long-read molecule technologies for assessing structural variant in the host genome, with the idea that it could contribute to COVID-19 susceptibility of progression, because we do know some individuals post-infection get symptomatic and definitely progress to ICU or death. The second objective was to create a global networks of lab geared towards data generation and data sharing, mostly focused on host genomes, structural variant genomic data. The third objective of our consortium was to form a global forum where we can share this data which was generated among us from the different genomic methods, different bioinformatic tools, and different methods of interpretation of host genomic data. So it's not just one lab or one method of sequencing or one platform of sequencing. The labs which are part of our consortium come from different backgrounds with the different bioinformatic tools together. Lastly, the idea behind this was to develop a global guidance on data and knowledge sharing, again, mostly focused on host genome structural variant. We started with one lab at a time, and at this moment we have 35 plus labs across the globe which are actively contributing to our understanding of structural variants in host genome for our individuals who are affected by COVID-19. Each and every lab brings in a unique understanding of their understanding and knowledge of COVID-19 to the consortium, and it has been extremely fruitful collaboration with all of these individuals come together. We meet once a weekly over a call, 11 a.m. on a Wednesday Eastern time. I think we're changing that time based on availability. And then that has resulted in a pretty significant amount of data generation and knowledge base on the COVID-19 host genome. And that's what we plan to discuss today. Our approaches to understanding the COVID-19 host genome, as well as our initial data. So I just want to give you an introduction to today's speaker lineup. We have Dr. Sivash from UC San Diego, which is going to talk about the bioinformatics tools, independent bioinformatics tools for identifying as well as analysis of structural variants in the whole genome. Dr. Silvio from West Virginia Commonwealth University, his idea about using bigger data in data mining bigger data, genomic data for identifying structural variants. Dr. Alex from Hoyshan from Radboud, he had a major publication on structural variants in Europe. Dr. Michael Zodi from New York Genome Center, Dr. Eric Jarvis from Rockefeller University, Dr. Alan Bex and Dr. Catherine Bronston from Boston Children's Hospital will talk about the MIS-C syndrome in children. And at the end, I'll wrap up with our data on COVID-19 host genome, especially the individuals who were sick, symptomatic sick, admitted to ICU and died. So stay tuned for the speakers to get started. Thank you.
Hello everybody, my name is Sivash Raisi-Dehkar, PhD student from UCSD. We developed a new tool for aligning the optical map data and the SV detection pipeline named FANDOM. FANDOM, or Fast Nested Distance Seeding of Optical Maps, is an optical map alignment tool and SV detection pipeline. FANDOM works based on the novel seeding algorithm to rapidly reduce the alignment search space. As you know, optical map data are high coverage, more than 100 or 200 of the genome, and aligning 2 million molecules would take more than 700,000 CPU hours. So having a very fast and robust alignment is very necessary. Our tool is an independent tool that works mostly for the Bionano Saphyr data, and on average, it takes 1.5 CPU seconds per fragment, and it takes 1 hour for the sample for each sample to run. There are two main. Our tool is faster than the existing tool like the Ref Aligner or the IronBlast. As the results show here, as you can see, the FANDOM is 4 to 6 times faster than the IronBlast and 13 to 14 times faster than the Ref Aligner. Also, for calculating the precision and the recall, we selected 207,000 molecules from each cell line. This cell line, Na12878, GM09888, and so on, as you can see their name here. But it is not trivial to designate an alignment as a correct or incorrect. So for detecting the true alignment, we align all these molecules with these 3 tools. And then we apply the majority rule. If an alignment at least reported by 2 tools, we assume it as a true. As we can see here, the precision and recall of the FANDOM is much better than the IronBlast, and it is really close to the Ref Aligner, but it is 13 to 14 times faster than that. So it is worth to use the FANDOM as an aligner and SV detection pipeline. Also, FANDOM has a very sensitive detection of the partial alignments that enables our SV detection pipeline to have a discovery in cancer cell line or other cell line for detecting the structural variation. In the Covet project, we use the FANDOM as an independent tool to kind of confirm all the finding by other tool, by the Bionano pipeline. As we can see here, this is the result of the fandom. This table is the table 2, which is the list of the unique structural variants overlapping with coding regions of the gene, which are identified in the patients with the COVID-19. As we can see here, for example, lots of unique structural variation that are detected by by other tools like the Bionano pipeline are also confirmed by the fandom. There are only 2 of them that were not detectable by fandom because the resolution of the finding, the inversion or deletion less than 30 kilobase per were not detectable with fandom. So more than 90% of the structural variation that were found by the Bionano pipeline are also confirmed and found by the fandom. Here is the table that all these green ones are detected by the fandom. And this is the continue of that number. All these 16, except 2 of them, are detected by the fandom. In the continue, I'm visualizing 2 of our findings and show how we detect them. Here is the example. This is the duplication that find in the chromosome 1 in the gene, in these 2 genes. As we can see here, also this visualization made by map optics. As we can see, there's one purple region here that is duplicated. There is 2 partial alignment for the contigs to the molecules. As we can see here, from the label 1350 to 1382 aligned to the purple region. Also, the label from 1386 to 1419 again aligned to the purple region. I have some schematic like that here to explain what is going to happen. As we can see here, these regions, the purple region, is got duplicated in the query, which is the signature of the duplication in our case. And this is the screenshot of the genome. As we can see, this region of the duplication contains these 2 genes that exactly we wrote their name here, HITER1 and EDART. This is the example of the another structural variation that were found by fandom. This is the duplication in the chromosome 19, the gene ZN443. Again, there is 2 purple regions that align into different region in the query, the context that we had. I have again the same schematic for that. As we can see here, these 2 green regions align to this purple region on the reference. So this is the signature of the duplication, and we find the duplication in this gene, in this chromosome. Here is the screenshot of the genome browser, and as we can see again, this gene is got duplicated. And thank you so much for listening to me.
I'm Silvio Alimbagano. I'm associate professor at the University, Virginia Commonwealth University, Department of Psychiatry, yeah? And I'll present today about using the existing C19 GWAS UK Biobank exome plus empirical transcriptomic studies when they will be available to prioritize sequencing SNVs uncovered using Bionano technology. The useful genetic study for C19 severe outcomes are COVID-19 host genetics, which is here is on the web available with results and all, where we have GWAS plus some of the post-GWAS type analysis like TWAS and other things. We have UK Biobank, which has exomes from 250K subjects, yeah, and some of them already had COVID. And this info can be used by us to prioritize Bionano SNVs in genes of interest, yeah? Now, using existing studies in prioritizing the genes are how we want to do it is first we will impute the C19 GWAS statistics, meaning the Z-score, to the latest reference panel. And our group has a 33K panel that will allow imputing Z-score for some fairly rare variants, even below 0.5%. Yeah. We might also get some interesting signals for certain rarer variants that can be followed up using Bionano technology. And afterwards, we apply the TWAS to imputed GWAS summary statistics of the more severe outcomes, yeah, like hospitalization that require ventilators. Afterwards, we'll get the gene level and pathway level p-values and z-score using our so-called JPEG mix tool, which is a TWAS for gene and pathway level analysis, pathway level being a novel area. And afterwards, we can also apply the theta type of methods to rare variants in UK biobank exomes for some of the more severe C19 outcomes, yeah? Again, that will be chosen there, maybe people that went to the hospital and were put on ventilators, yeah? For this one, we can obtain by gene and pathway p-values similar to the common variants, yeah, from the C19 host genetic. And afterwards, we can combine the common from GWAS and TWAS with the rare variant statistics from theta. And when available, we can also put empirical transcriptomics studies available. And by looking at the concordance between these statistics, we can obtain the combined common and rare variant statistics that can be used to assign prior probabilities to certain genes, for instance, those that are finding some of the Bionano SNVs from our consortium. So how can we use the prior probability, yeah? Info in prioritizing SNVs for the follow-up. First, we can prioritize those SNVs near or within genes with higher priors. We can prioritize those SNVs near or within genes from pathways that have higher priors, yeah? And subsequently, when the sample size is large enough for our consortium in thousands of patients, we can compute a by gene and pathway statistics from our Bionano sample, and we can combine Bionano sample stat with those from GWAS and UK Biobank, plus, as I said, empirical transcriptomics when available. And in this way, we can test for concordance, and we will choose those SNVs that will have SNVs genes and pathways that have the highest concordance. I'm not working alone. I have collaborators at Virginia Commonwealth University. They are Brian Riley, who is a molecular geneticist, yeah. Huang Nguyen, he's a specialist in so-called theta that looks at the rare variants by gene statistics. And Ken Candler, who is a well-known genetic epidemiologist. Thank you very much. Bye.
Hi there. Thanks for joining today's seminar series. My name is Alex Hoyshan. I work in Nijmegen, the Netherlands, and I have a great interest in the effect of rare genetic variants on severe forms of COVID-19. Just to introduce myself and my group that I had here in Nijmegen, my group is called Genomic Technologies and Immunogenomics. So I have a core interest in developing and applying genomic technologies for my own research questions that really focuses usually on rare diseases. But then together with my colleagues, I also try to push latest technologies towards diagnostic use, specifically for optical genome mapping. We're using the Bionano system already a little less than 2 years by now and have processed more than 250 samples since then. Next to this more technology-driven interest, I really have a core interest since several years now in immunogenomics, and they're mainly a rare disease focus where we try to study new and novel genes for inborn errors of immunity and primary immunodeficiencies. And again, also here, I collaborate closely with my colleagues in diagnostics, where usually cases that are analyzed for diagnostic exome sequencing with a suspicion for a severe immune defect, my group comes in to reanalyze the data and perform a research follow-up on novel findings. Some of that process that really focuses on exome sequencing here has been described by my previous PhD student in this overview article, if you're interested. This is just to tell you that we already joined the Cytogenomics series with 3 other presentations, one by our wonderful collaborator Leila El-Katabi, one by my former postdoc Tuomo Mantere, and I also myself gave a talk already on research findings using optical mapping. The reason why I also joined the Covet SV consortium is our most recent findings from this summer for very severe forms of COVID-19. So with an entire research team here in Nijmegen, we discovered 2 families in which two brothers each developed extremely severe forms of COVID-19. Sad enough, one of the 2 brothers of family 1 even passed away due to the severity of the disease. And what is truly remarkable about these 2 families is that all 4 brothers are extremely young. They are 31 and 29 years of age in family 1, and even younger, 21 and 23 years of age in family 2. By performing diagnostic rapid clinical exome sequencing, we identified rare loss of function variants in a gene called TLR7, which encodes for the toll-like receptor 7. In addition to identifying these mutations that you can see in the cartoon on this slide, we also have shown that the individuals have a defective interferon signaling due to their loss of function mutations in TLR7. And as such, we were able to show that TLR7, once defective, really leads to a downstream defect in mainly type 1 and type 2 interferon. Furthermore, this also implies the TLR7 pathway as an important innate immune response pathway to battle off SARS-CoV-2. Interestingly, our finding was just replicated by an Italian consortium who identified rare TLR7 variants in 6 out of 156 individuals, all male individuals, obviously. And this consortium was also able to show a similarly impaired type 1 interferon defect. This work is also very much in line with the very recent publication by the COVID host genomics consortium led by Jean-Laurent Casanova and an entire team. This group interestingly identified severe loss of function variants in the more extended type 1 interferon pathway, and mainly genes that have been previously implicated in severe viral pneumonias. And they found a total of 3.5% of severe COVID-19 cases that potentially could be explained, or their severe predisposition to COVID-19 could be explained by these type of variants. So seemingly type 1 interferon is truly important, and now there's efforts ongoing to really understand what that would potentially mean for treatment of individuals. With this interest in Bionano optical mapping and an interest in severe forms of COVID-19, I joined the COVID SV consortium simply because I believe that there must be more rare genetic host factors out there that could lead to severe forms of COVID-19, and they could be very helpful to identify. We may speculate that that again may be related to a functional defect in interferon production. And for those working on more common diseases, I think it's important to reiterate that some of these genetic defects that we identify are individually extremely rare, but collectively they could already explain a certain percentage of cases. Furthermore, these rare disease findings and severe predispositions may allow important insights into the diagnosis, disease mechanisms, and biology in general, and may in some cases even offer novel treatment options. To my understanding so far, there have not been very systematic assessments of structural variants as an important cause or important source of genetic variation. So therefore, I got this interest in the Covet SV consortium. And I think it's good to point out again that structural variants itself may have a greater a priori chance to be rare and disruptive in the human genome. Furthermore, I think it's important to keep in mind that occasionally point mutations and structural variants affecting the same genes or loci may actually very much complement each other. Especially for extremely rare events, there would be a lot of value in looking at all types of variation. So with that, I hope also the input from my group can add to this SV consortium, and hopefully we can identify novel insights into this devastating pandemic and disease. The work I've been involved in COVID-19 was obviously teamwork from a lot of people here in Nijmegen, mainly driven by my highly talented PhD student, Cas van der Made, heavily supported by my colleagues at Internal Medicine, Frank van der Verdonk, and certainly also my mentors, Han Brunner and Mihai Niteer. We're very much looking forward to see you at the roundtable discussion and answer any questions that remained after this presentation. Thank you very much.
Hello, my name is Eric Jarvis. I'm glad to be part of this symposium and presenting work that my lab has been doing on COVID-19 research in relation to genomics. And just to give everybody a little bit of background, I'm actually a neurobiologist and a genome biologist, and I am running the Vertebrate Genomes project. I'm chair of it, whose mission is to generate high-quality genome assemblies eventually of all vertebrate species. Right now we're doing all vertebrate orders. And with the ability to produce high quality genome assemblies, I got involved in this structural variation consortium with the idea to find out whether or not we could find greater structural variations in people who have severe COVID reactions compared to those that do not. And to use that technology to address this question in collaboration with Bionano and others. And today I'm going to tell you about a story on the interferon 1 alpha and beta receptor 1, that there's some variation in that receptor, we think, some preliminary findings that correlates with degree of COVID-19 severity. So the data set that we're working with actually comes from Bionano, the initial data set, a metadata approach. Here we have hospitalized patients, those who have been severely ill in ICU or have died, 30 of them, and controls, a random set of controls that are in the Bionano collection, 57 individuals. Bionano generated chromosomal length Bionano maps of these persons, aligned it to the human GRC H37 reference genome, and called variants for each haplotype, not just one haplotype, but for both haplotypes throughout the genome. And that's important to focus on both haplotypes. And initially, just looking at these variants in the hospitalized patients, we found some interesting genes that are highly represented with more variation than you would expect by chance in some immune genes and respiratory system genes, and actually even genes that are involved in coronavirus interferon induction. At a significantly high level of enrichment of gene ontology categories. But when we looked at the controls, we actually found some of the same gene families that were showing greater variation than usual, than you would expect by chance, and also in the controls. And that then led us to realize, well, these genes, including immune-related genes, are genes that actually do have a lot of structural variation in them to begin with. So we can't use this as a criterion as compared to the rest of the genome background to ask if there's a difference between the severely ill patients and those who in the general population background. But we did find something else interesting when we started to look at the actual heterozygosity. And that is for this interferon alpha and beta receptor 1 subunit, we found that there's some variation in the upstream promoter region of this gene in terms of insert size. And we found that the control group, roughly 20% of them have heterozygous, large heterozygous variation in this region. Whereas in the hospitalized group, an amazing 78% or so of these 37 patients had high levels of insert size heterozygosity in this region. Whereas in contrast, another gene involved in the immune pathways, interleukin-1 receptor type 1, where both roughly 60% of the control and the hospitalized group have heterozygous variation at a similar level as what we're seeing over here for this interferon alpha beta receptor. So, what this meant is that there's something different, not about the actual individual the individual variant, but the level of heterozygosity of that variant. So we actually then aligned the Bionano maps back to the assembly and try to figure out what exactly is that variant that we're seeing. And what we're seeing is that there is this highlighted in this red circle here, in front of the 5' region of the gene, there is a repetitive, a tandem repeat structure of nucleotides that are varying from one person to another and also one haplotype to another. And what we see with the patients who become severely ill is that the variation of those two different haplotypes in this tandem repeat is greater. And we can map that out to show here that there's variation in the length of this tandem repeat when you add up both haplotypes together. But and you can see a difference here between the control and the severely ill patients. And so what we're going to do, and so putting all this together, going back to this plot here, is what we think is going on is that the greater heterozygosity might be causing a deficit in the function, let's say, of the regulation or the function of the first intron of this receptor, that in its more heterozygous state, where the two different variants are more similar to each other, is not as detrimental to, let's say, the function of this gene. And so this is consistent with this interferon gene pathway being found involved, or at least other genes like the interferon molecule itself from the Casanova group and the consortium that he's helping to lead, that different genes in this same pathway are being found to be associated with the degree of severity of this disease. And we think this is one more example. And to show you in a cartoon fashion, this diagram from Vertebrate et al. Shows that when the virus binds to a cell, it binds to the ACE2 receptor, it gets taken up, that causes an immune response in that cell to release the interferons. They bind to these receptors that we're finding this difference in heterozygosity, and recently also the interferon alpha-2 receptor, a publication in Nature a few weeks ago showed that there's some variation there that's associated with degree of severity. And these receptors can dimerize here. And we think that the heterozygosity is affecting that level of dimerization to cause a differential immune response in cells that respond to the infection. So future efforts, what we're going to do is we're going to generate more complete de novo chromosomal level reference genome assemblies of COVID-19 patients, 20 persons who have been severely ill, 20 controls who are asymptomatic individuals matched for age, sex, and pre-existing conditions. And we're going to use the vertebrate genomes pipeline for this, where we have PacBio long reads, Bionano optical maps, and high-C data from various groups like Genomics and Dovetail. And we're going to use these data types to produce as complete a genome assembly as possible, and then interrogate this region of the genome to find exactly what kind of variation we're seeing in heterozygosity and other types of variants. And we're going to compare the human data with data from 260 vertebrate species that were generating high-quality data for representing all vertebrate orders, including species that are more susceptible and less susceptible to being infected by COVID-19, to see if there's any correlation with humans who show greater susceptibility to being sick from this virus. We're going to do that from the nucleotide to the structural level of analysis on human and non-human animal genomes, and identify the exact kind of structural variation we're seeing in these cases that I just showed you in the previous slide with the IF and RI1 gene. And if you want to join this effort, I encourage you to contact me. And before I finish up, I'm going to go right back to the beginning to thank my colleagues here, Farouk, who is a graduate student in the lab, who did a lot of the analysis, Olivier Fedrigo, who helped also with the analysis, who's a Bionano expert in our lab, Alex Hasty, who works with Bionano, who also helped us out with these analyses, and myself, who's running or supervising this project. And I will go ahead and stop there, and thank you for your attention.
Hi, I'm Michael Zodi. I'm the Scientific Director of Computational Biology at New York Genome Center. I'd like to start by thanking the organizers for giving me the opportunity to speak to you today about our COVID-19 genomic research at NYGC. I'll start with an overview of the Genome Center's COVID-19 research activities. In the early part of 2020, when New York City was the epicenter of the COVID outbreak in the United States, we established the COVID-19 Genomic Research Network with the goal of trying to unify COVID-19 genomic research in the New York tri-state area and provide support from NYGC for genomics-related research for all the New York and New Jersey institutions that were interested in participating in that. Since then, we've hosted a bi-weekly large meeting with presentations and discussions of current research in COVID-19, and we've also been working with our partner institutions to generate genomic data that we hope can enlighten the struggle against treatment or for treatment of COVID-19 and understanding of the spread of the disease and the variability of severity in different patients. As part of our efforts, we've undertaken a wide range of genomic activities, including using our New York State approved clinical lab to do COVID-19 testing. We've also been doing full-length SARS-CoV-2 viral sequencing with a number of our hospital partners in the New York and New Jersey area. And we've been offering low-pass sequencing of COVID-19 patients to do wide-scale imputation as an alternative to microarray-based genotyping for wide association studies of host risk for severe COVID-19 or for COVID-19 infection in general. And our faculty labs in particular have been working on several areas of functional genomics of the SARS-CoV-2 virus and its interaction with host cells and the proteins both on the viral side and the host side that are crucial to that. But the piece of this that I'm going to talk about mostly today is our efforts of whole genome sequencing on severe COVID-19 patients. And so what we've targeted to do is to provide whole genome for severe cases of COVID-19. And so we define this as patients who have severe disease, but who are without prior known high risk or prior morbidity that puts them at risk for severe COVID. So we broadly define this as individuals less than 50 years old with no known prior conditions such as diabetes or hypertension or obesity that we know independently predispose people to more severe cases of COVID. And these people wind up in the ICU and or being mechanically ventilated. We've also expanded this last spring after the discovery of what's now known as MIS-C, or Multi-Inflammatory Syndrome, in children, to include whole genome sequencing of children who are affected by this subsequent to a COVID-19 diagnosis. And so the reason that we lean on WGS for this is to identify rare variants that wouldn't be found by a chip assay that might be involved in particularly severe cases that we expect that this highly selected population might be enriched for. Also, to be able to assay variants in regions that are difficult to assay by less comprehensive tests. So, for example, being able to do detailed HLA typing from the whole genome sequencing that might not be possible from less comprehensive assays. And of course, to be able to look at structural variation that may be playing a role in susceptibility or severity of COVID-19 infection. And a lot of this work has been done together with Jean-Laurent Casanova at the Rockefeller University and his international COVID human genetic effort. And there are two papers that were released on that last year that included some data and work from the Genome Center, one of them identifying rare variants conferring inborn errors of type 1 interferon immunity in patients with severe COVID, and a second one identifying autoantibodies against type 1 interferon in patients with severe COVID. And so that's an example of the kind of work that we're doing and what we hope to accomplish with whole genome sequencing. So to get into a little more detail of our work with the COVID-19 Host Genome Structural Variation Consortium, we're hoping to do whole genome sequencing of severe patients who've also been characterized by Bionano by the Host Genome Structural Variation Consortium. And structural variation from whole genome sequencing is something we've been deeply interested in the Genome Center, even predating COVID. And so we have an extensive structural variant calling pipeline that involves standard methods for detecting things like deletions and insertions relative to the reference genome, copy number changes. But we also have an ongoing program to develop a novel method that we call Absinthe for detecting large insertions relative to the reference genome using short read sequencing, which has historically been considered to be very challenging to do. But we've shown that we can detect very large events and cleanly identify them and then genotype them from the short read sequencing. We're also interested in mobile element detection, and we've done a lot of work in looking at microsatellite expansions as well. And so in collaboration with the consortium, our hope is that by applying whole genome sequencing to these individuals, we can provide refinement of the breakpoints of events that are detected by Bionano, as well as allowing that greater refinement to let us say more about gene and functional element overlap of these events and try to get into potential functional roles of them. The whole genome sequencing also allows us to identify some smaller events that are less easily detected at the resolution level of optical mapping and add those into the studies. And then lastly, one of the other things that we've been working on for a while now at the Genome Center is the imputation of common structural variants for use in association. And I want to say a little bit about that. We've developed methods to take the whole genome shotgun, phase structural variants together with single nucleotide variants, and build imputation panels that include both structural variants as well as single nucleotide variants. And the idea is that by imputing these into large genotyping studies, we would be able to conduct direct association of structural variants. And so to take these elements that are difficult to identify in genotyping studies and make them available for direct ascertainment in studies using genotyping data. And I'm showing a little bit of data here from some of the work that we've done on 1,000 genomes, building combined structural variant and SNP panels and imputing these, and showing that really the info scores that we get from imputation of structural variants are pretty much equivalent to the ones that we get from single nucleotide variants, and that when we assay this against a gold standard set where we have multi-platform ascertainment of structural variants in a specific sample, that our genotype concordance for imputation of these from an unrelated panel is really quite high and quite comparable to what one would expect to be able to do for single nucleotide variants. So we're also excited about using these techniques to take common variants that are discovered out of the consortium and try to move these into some of the larger genotyping-based studies that have been put together for looking at COVID patients as well. And so I'll stop there and just briefly acknowledge all the people at New York Genome Center that have been involved in this project in particular and in our COVID work more broadly, as well as the individuals that are part of the CHGE effort and some of our partner hospitals in New York City and New Jersey that have been providing us with SARS-CoV-2 and COVID patient samples for the work that we're doing. Thank you.
Hello and welcome. My name is Alan Beggs. I'm director of the Manton Center for Orphan Disease Research, and together with Dr. Catherine Brownstein, we direct the Molecular Genetics Core Laboratory here at Boston Children's Hospital. We'd like to tell you today about the research program we're developing with the COVID-19 Host Genome Structural Variation Consortium to understand the contribution of genomic structural variation to disease severity in children. So the first point I'd like to make is that COVID-19 is a disease of all ages. If you look at these graphs of age related to either diagnosis with COVID-19 on the left or death on the right, based on data from last week on the CDC website, you'll see that as many as 10% of COVID-19 patients are children under the age of 17. Fortunately, the mortality rate in this group is quite low, less than about 0.2%. However, even these deaths are important to understand. In contrast to the 0.2% rate of mortality among patients with COVID-19, a new syndrome called Multi-Inflammatory Syndrome in children has recently been identified as having a significantly higher mortality rate and being medically and biologically distinct from primary COVID-19. This is a syndrome that was first recognized in the United Kingdom in late April of last year, in which 8 cases, including 1 death, were reported of previously healthy children who had PCR positive COVID-19 and presented either during or soon after their COVID-19 disease course with hyperinflammatory shock and features that were similar to Kawasaki syndrome. By the middle of the next month, in May of 2020, the New York City Department of Health had identified over 102 patients with what they recognized to be a multisystem inflammatory syndrome that was associated with either prior or current SARS-CoV-2 infection. By the middle of May, the CDC had actually defined this condition in a health alert as a condition affecting individuals less than 21 years of age, presenting with fever, laboratory evidence of inflammation, typically evidence for 4 or more inflammatory biomarkers, and evidence of clinically severe illness, typically requiring hospitalization, with multi-system organ involvement, often involving either the heart, renal system, respiratory, hematologic system, GI, dermatologic, or neurological, and no plausible alternative diagnosis. In addition, to meet criteria for definition of a case as MIS-C, children were required to have be positive for either current or recurrent recent SARS-CoV-2 infection by RT-PCR, serology, or antigen test, or a known COVID-19 exposure within the 4 weeks prior to the onset of symptoms. Very rapidly, within the next month or so, at the end of June, 2 landmark papers were published describing syndrome in almost 300 cases from across the United States. On the left, paper from colleagues based on a multi-center study called Overcoming COVID-19, funded by the CDC and led by investigators here at Boston Children's Hospital and Dr. Adrian Randolph, who's a member of our research team. And on the right, a study similar size and scope from the collaborators with the New York State Department of Health. So these two studies really kind of helped put MIS-C on the map and define what it was. To understand MIS-C, it's important first to distinguish it from acute SARS-CoV-2 infection leading to COVID-19. So as in adults, COVID-19 in children has an unpredictable clinical course with an extremely wide clinical variation, ranging from asymptomatic carrier state to a self-limited upper respiratory infection, including mild or moderate disease treated on the wards, and at the extreme end of the spectrum, severe or critical disease that often at that point requires mechanical ventilation and or other life support. In contrast, MIS-C is really a post-COVID-19 syndrome, characterized, as I've said, by multisystem inflammatory findings affecting multiple organs, and typically occurs within 2 weeks to a month after infection. So in these two New England Journal studies, as many as 92% of children had gastrointestinal involvement, cardiovascular involvement in about 80%, hematologic, clotting, mucocutaneous, or respiratory involvement in about 3/4. And interestingly, as many as 40% had Kawasaki-like features involving the heart, and 8% developed detectable coronary artery aneurysms, leading to potential life-threatening complications. Biochemically, more than 90% had more than 4 elevated inflammatory biomarkers, thus meeting CDC criteria for this diagnosis. And the median hospitalization was a week, with about 80% requiring intensive care, 20% requiring mechanical ventilation, and as I said at the outset, a much greater mortality rate than COVID-19, with as many as 2% of children dying. The average age was 8, and in the New York City cohort, the 40% of children ranged in age from 6 to 12 years of age. So this is really a syndrome of children, although recently reports have been coming out of occasional adults with similar clinical presentations. Interestingly, in contrast to Kawasaki syndrome, which affects primarily children of Asian descent, there was no racial or ethnic predilection among children developing MIS-C. And treatment was supportive as well as directed towards immunomodulation, with as many as 3/4 of children receiving IVIG, half of them receiving glucocorticoids and IL-6 inhibitors, and another 20%. So with that, I'm going to turn it over to Dr. Brown's team to tell you a little bit about the resources we have here at Boston Children's Hospital in our project with the consortium, and tell you a little bit about our experimental plans.
Boston Children's Hospital is a 400-bed facility with 25,000 inpatient visits and over 500,000 outpatient visits per year. We are a regional referral center for New England and an international tertiary center. As of January 7th, Boston Children's Hospital has tested 42,395 patients for COVID-19, of which 1,034 were positive, and has reported 67 cases of MISC. Dr. Adrian Randolph leads the CDC-funded Overcoming COVID-19 multi-center surveillance study, and together with Dr. Janet Chow, lead Taking on COVID-19, an international COVID-19 patient registry, biobank, and natural history study. In June, Dr. Randolph led one of the first comprehensive studies defining MISC and describing its natural history in 186 patients from 53 hospitals in 26 states. The Children's Rare Disease Cohort Initiative, led by Drs. Piotr Slitz and Shira Rockowitz, working with the Hospital Institutional Centers for Clinical and Translational Research, banks specimens and has generated short-read exome and genome data on COVID-19 patients as part of taking on COVID-19. Our hypothesis is that specific environmental, medical, or genetic factors may predispose some children to severe outcomes of SARS-CoV-2 infection. Genetic factors may include both single nucleotide variations and large-scale structural variants that are not detectable by traditional next-generation sequencing techniques. Therefore, our experimental plan is to have strict inclusion criteria, including a positive RT-PCR or serological testing for SARS-CoV-2 infection, an age less than 21, and informed consent for taking on COVID-19, overcoming COVID-19, or the Manton Center for Gene Discovery Core IRB protocols. We plan to enroll 50 patients each with MISC, severe COVID-19 requiring hospitalization, and asymptomatic or mildly affected SARS-CoV-2 positive patients. We will query them for structural variant detection by optical genome mapping on the Bionano Saphyr and short-read Illumina-based exome or genome sequencing. This work will provide the opportunity for follow-up, RNA-seq, or other RNA-based, expression-based studies. We will have data sharing and joint analysis with the COVID-19 host genome structural variant consortium. And hopefully next time we speak, we'll have exciting results to share with you. Thank you.
Hello, everyone, and welcome back to my talk. What I'm going to summarize today is our data information, what we're doing. We have captured in the last few months on structural variant analysis by optical genome mapping in severe COVID-19 patients. Again, I'm Robinhood. I'm a pathologist at Medical College of Georgia, Augusta, at Augusta University. And this is a collective work through our COVID-19 Whole Genome Consortium and their members. These are my relevant disclosures associated with the content of the talk. Most importantly, the slides, the opinion expressed here are my personal opinions, and they do not represent the opinions of my collaborators or the employer. And the Saphyr instrument, which I'm going to mention in this talk, is intended for molecular biology applications and not intended for diagnosis, prevention, or treatment of the disease. So some background on why we are doing this, as you guys must have seen through the presentations, not only for today, for the entire week, that the structural variants, including the CNVs, accounts for a significant portion of variation among individuals. And these variations do play a significant role in host immune response, and this is very well documented, especially the large SVs. When we started looking at structural variants and the available data for COVID-19 predisposition, we felt that the existing consortiums were mostly focused on sequencing, and which ideally would miss some of these larger structural variants. And to investigate these host genomes, especially in the individuals who are severely ill at very large structural variants, we put together this consortium of groups and labs and started discussing what are the platforms, what's the ideal way, what should be the population in which we should be looking at. With one of the most important goal is, why do some people get sick and some people end up being ICU and dead with the viral infection? Is there something in our host genome, especially at the large structural variant, we can identify? So that this is our overall goal for this study. The way we designed this was, at this current moment, we have 37 individuals who were infected with the SARS-CoV-2 virus and had the COVID-19 disease. They were admitted to ICU, and the control group we have is two. One is the Bionano control group, as well as we have individuals who are infected by the SARS-CoV-2 virus, but they're absolutely asymptomatic. So those are the control groups we're using in this study. In this demographic, I mean, the inclusion criteria was anyone who's confirmed with a SARS-CoV-2 PCR test and would require for a mechanical ventilation or at least a fractional inspired oxygen of 60% or more. The individual we were able to include in the study ranged from the age range was 19 to 83. Good mix of male to female, dominated by African American individuals because of the population around our state. And we do have a decent mix of chronic pre-existing condition in these individuals, mostly diabetes, hypertension, chronic kidney disease, and asthma. The platform which we use for my presentation is Bionano Saphyr system, and the de novo assembly pipeline which was used was the human genome 19 reference genome. I'll jump onto the results. We identified unique 11 structural variants among 38 genes, out of which 7 were rare and unique, and 4 were common structural variants. Most, if not all, the information is extensively discussed in this non-peer-reviewed preprint on medRxV. If you feel, please go ahead and look into and let us know what you guys think about the data. So this is the structural variant number 1, which includes the deletion in the exon 3 and 4 of the gene DPP4. Here we show that this particular deletion in exon 3 and 4, and as you know, DPP4 is an immune gene which is implicated in diabetes and the response to diabetic drugs, and we're able to show the dosage of this deletion with expression using control DNA. The second structural variant is on the X chromosomal, which is basically a duplication involving the STK24. And here on the chromosome X, we can see the duplication. And again, the STK24 is associated with innate immunity and inflammatory response. And these are the dosage studies which shows the expression to show the duplication and increased expression. And continuing on STK24, when we looked at all the individuals, especially the 11 asymptomatic and 12 severely ill individuals with normalized RNA, you can clearly see there is a significant fold change in individuals who are infected with SARS-CoV-2 and absolutely no symptoms versus individuals who are infected with SARS-CoV-2, end up with a COVID-19 disease, as well as had to have assisted oxygenation or ventilation. And as you can see, one of the cases which we have a highest STK expression had a partial duplication of STK24 gene. So this basically proves what we were seeing on the optical genome mapping on Saphyr pretty much has a functional approval in terms of expression of these. The structural variant number 3 was involving the deletion of the ZDHHC1 gene. It is a 146 kb heterozygous deletion in chromosome 16 that contains up 6 genes, which includes the PKL HG4 KCD19, LRRC36, TPP3U1, and the ZDHHC1 gene. And we were also able to show the dosage experiments with the control to show the deletion has the functionality or the functional studies of that deletion are there. So these are the 3 unique and the private events which we saw, I think, were clinically important. We are studying them extensively in different ways. So to conclude, the optical genome mapping definitely identified structural variants in 3 different categories. One is the innate immunity and inflammatory response, and it has a group of genes which are very well studied, not only in SARS-CoV-2, but also the SARS-CoV-2-1 and another viral infection and their inflammatory response. The second group which we identified, the structural variants, was yearly resistance to pathogens, especially the mucus secretion, and the MUC4 gene had very well documented structural variant in that particular gene. And the third group, which we have pretty significant structural variants, was viral replication, spread, and RNA editing. And I think this group, we are exploring it further to see what are the implications of this group in post-infection, not just making it asymptomatic. So our overreaching goal for the consortium as well as my lab is to recruit 1,000 individuals with a significant number of asymptomatic individuals as a control, continue functional studies for DPP4 and STK26, because these were the early insights or first insights in these structural variants in COVID-19 individuals. We're also working with our collaborators in the consortium to do a targeted sequencing for TLR7 gene and the downstream pathway. We are also investigating these structural variants and where do they are in the GWAS studies or the existing data, as well as some emerging data on the sequencing. We're also doing some parallel whole genome analysis on our subcohort. And then we do also plan to do comparative genomics with other vertebrates with the collaborators in our consortium. So this is our proposed diagram, which shows a key viral host interaction pathways highlighting genes that we found to have structural variants in COVID-19 in our patients. I know this is early data, but if you look at the upregulated and downregulated and the balance between them and the structural variants identified. So this is our proposal, which is still a work in progress. Again, this is a part of that presentation. This is part of the paper, which can be looked at it more in details. Again, as I mentioned earlier, this is a very collective global effort to put and collect this knowledge base for all of us through the COVID-19 host genome structural variant consortium. We have a variety of individuals with expertise in different areas which are helping us put this together. And I would like to help Dr. Sejpal and Dr. Mondal in my lab, and from Bionano Genomics, Dr. Clifford, Dr. Pang, Dr. Hasty, Dr. Chobe, Jeff Robinson, and William Bano to help us put these, not only the consortium, but the data and everything together. Thank you.
Hello, everyone. Okay. Let's get all our expert panels on. Let's see. Can you guys hear me?
Yes.
Yes. It looks like we are getting all our experts in. Welcome everyone to our live Q&A and our expert panel session on day 5 of our symposium. And as I mentioned earlier, this is actually a little out of the theme of our next generation cytogenomics symposium. So this is a bonus day for all the attendees. And I have to acknowledge that when I was interacting with Ravi and talking about what our plans are for the symposium, to be happening on 4 days, Ravi said, well, why don't you have a COVID-19 bonus day, 5th day? And that is how this all started. So I do want to welcome everyone, and I'd like to open this live Q&A and an expert panel discussion with all of you, starting with Ravi. So Ravi, I'm going to start with you so that you can take it on and basically, first of all, tell all the viewers that with this COVID-19 pandemic, a lot of the speakers have already alluded to the differences between the severe and the asymptomatic cases, but why is this important from testing and other aspects of this pandemic?
Thanks, Alka. We still getting echo?
No,
Okay.
we're good. Yeah, we're good. Yeah.
Thanks, Alka, for putting this thing together. I think I do understand this is a next generation cytogenomics symposium, but you can't just shy away from COVID-19 as each and every one is dealing with it. So we all appreciate you and your team to put this thing together. And I think we were able to present some of our ideas, our initial data, what we are doing here, and that has really came out to be really nice. So going back to the question, and I think that question probably is the origin of what we are doing, as well as why we end up being a part of this consortium and spending our time doing research for this particular entity. So as you know, millions of people, 250,000 people in the US are getting infected on a daily basis now, but we know a very small percentage of them are symptomatic, and that to extent very mild symptoms. And in that, a very small percentage of symptomatic individual are getting need some oxygen support or getting to ICU, and then again, very small percentage of them end up dying. But even if this is a small percentage, the numbers are pretty high because pretty much every other individual is getting infected in the community, so the numbers are going to be high, and that's one of the reasons we want to identify, is there something in the host genome which we can identify if somebody's going to be susceptible for the infection or being symptomatic. So that's the whole background. And then coming back to the question about how do we define symptomatic versus asymptomatic, so I think asymptomatics are pretty straightforward. The definition of symptomatic is changing. The original definition was based on our understanding of flu, and it's symptoms of the flu associated with what we have been seeing in the past decades. Currently, I think it's most, if not everything, associated with the flu, but the key thing is you need a PCR positive test to call it symptomatic. But the key differentiator from symptomatic versus individuals who end up needing some kind of oxygen support or ICUs, I think that particular definition is changing, and that has nothing to do with the severity of the infection or response. Unfortunately, it's getting to the stage of availability of rooms with oxygen and ventilators in the hospital. So earlier, even if you have a small discomfort, you are asked to put with an oxygen support. Now I think that threshold is increasing, and I know it'll keep on increasing. So I think, at least in our study, we came up with a cutoff of 60% oxygen requirement as assisted oxygenation, and then obviously ventilation has its own criteria. So sorry for the long answer, but I think it is critical to understand these are moving posts and how we are dealing with this on a daily basis. So I would like to continue the discussion with my collaborators on here. I'm gonna start with Dr. Zodi, Mike Zodi. An amazing presentation, Mike. And we, again, coming back to Alka's question about the criteria we want to include in your study or our study, I saw you mention your earlier criteria. Can you just summarize those? And is there a chance you guys are going to go back and revise that or expanding that criteria for inclusion in your study? Mike?
I think we might have some issues. Mike, we cannot hear you.
Maybe, Mike, are you on mute?
Ravi, why don't we move on? And then when Mike comes back on, William, will you please send Mike some text about what we can do probably?
Okay, no problem.
Yeah, we'll come back to Mike again.
Back from Mike, coming back to Alan and Catherine, I think very similar theme of questioning. I mean, everyone is now interested, especially with the newer vaccine trials, which includes younger individuals. So how are you defining the criteria for MISCs at the Boston Children's? And well, what are your inclusion criteria in the study which you're doing there? Can you elaborate that or summarize that for us? Thank you.
So I'll say at the outset that the clinical work is being led by Adrian Randolph, who is the PI of the CDC-funded Overcoming COVID-19. And within Children's Hospital, we also have an internal study called Taking on COVID-19 that is a combination registry, specimen data bank, and natural history study. And working together with Janet Chow in our immunology department, who's doing a lot of that work. So I can't answer specifically your question, but in general terms, the clinical criteria that are being used, I believe they're following basically CDC criteria, which is, and as I mentioned in my talk, an age of less than 21, evidence of multi-system involvement, and laboratory evidence for elevated markers of inflammation, at least 4 typically. In terms of, you mentioned the vaccines, I think as you know, they haven't been tested in children yet. And so I believe it's slightly different age cutoffs for the Pfizer and the Moderna. One is 16, I think, and the other maybe 18. So they're not going to, the children, unfortunately, here at Boston Children's Hospital, many of our patients are not yet eligible for vaccination. But Children's, I should say, has not had an enormous rush of inpatients. Our inpatient load of COVID positive patients has been relatively manageable to date. We're in fact potentially taking patients from other hospitals in the Boston area to free up ICU space in those hospitals. So the COVID-19 cohort that we'll be studying is a combination of patients who've been referred to and admitted to Children's, and also ascertained through the National Overcoming COVID-19 Consortium.
The only thing I would add is that we are, well, I've heard that we are adding to the protocol the ability to enroll patients who get the vaccine once it's available for children and go on to get COVID, and also patients that have a reaction to the vaccine.
Thank you, Ravi, Alan, Catherine. And I think Mike is going to try and join by another device, and we'll come back to him. Alex?
Really appreciated all the presentations today. I have a question for Dr. Silviu Bacana. I really enjoyed your presentation, and I was wondering if you could please discuss in a little bit more detail about how GWAS can or cannot be used to find and analyze structural variations, and if there is some history of GWAS being used for structural variation analysis.
We are having some issues hearing you.
Can you hear me now?
Very faint, it's very faint. I apologize. I mean, this is live Q&A. We've had bandwidth issues pretty much on every day except one day where it was a complete success. So, all right, Sven, let's move on and see and revisit this question.
Yeah, of course. We have a question for Dr. Coley and Hoyshan. Yep. I'll just keep going. We heard about these toll-like receptor genes a few times now in several of the talks, and interferon as well. Can this knowledge be used in treatment at all? If these type of variants are detected quickly, can people be treated appropriately while they're in the ICU? Maybe Ravi, if you would like to go first.
Sure, thanks, Sven. I think we are very early in TLR's pathway, but definitely what Alex and his group has done and a couple of other groups in Europe have done is pretty significant information. But I still think it's early to get some targeted therapy against that. But I'm going to let Alex expand on that because he's on top of TLR. Alex, you want to take this?
Yeah, I'm very happy to add to that. So, I mean, I'm not an immunologist, but rather a geneticist. But my colleagues at immunology are really convinced that in people that are TLR7 deficient, in whom we already were able to show that they lack a severe and strong interferon response to the virus, they would be relatively straightforward to supplement with interferons. And interesting enough, in other large severe COVID cohorts, interferon supplementation is already tested in clinical trials, irrespective of the deficiency. So if it's efficient, then probably it's even most efficient in the patients that show deficiency for interferons. And on top of that, I should add that just, I think it's 3 days ago, the group of Jean-Laurent Casanova has for the first time treated severe COVID-19 patients that suffered from TLR3 and IRF3 mutations. So again, linking to the TLR and the interferon pathway, and they did so successfully with certain types of interferons. So I think in that sense, it's very promising. However, I also would like to add that, I mean, for the TLR interferon related structure variants from this very consortium, I think we still have to deliver several levels of evidence that they really suffer from a deficiency or a true genetic defect that for example, would lower their levels of interferon. And I think that's has not happened yet. But still, it's very interesting to see that potentially we find these targets.
Thank you.
Thank you, Ravi. Thank you, Alex. Our next questions are for Alan and Catherine again. So one of the questions is regarding MISC. A high percentage of children have GI involvement. So were any other sample types other than nasopharyngeal swabs, or what were the sample types essentially tested or will be tested for this study?
That's a great question, and I'm embarrassed to say that I'm not sure.
Okay.
I would say that one place somebody could go to look might be the clinicaltrials.gov entry for overcoming COVID-19. And I will find out from our clinical collaborators, and if there's anybody in the audience who would specifically like to know, feel free to look us up, find my email address, and I will try to answer their questions. I do know it's a variety of different specimens, and it's a very important point that certainly we're going to want to correlate RNA-seq results together with the constitutional genomic structure and sequences, and so there will be a variety of tissues that are sampled.
Thank you, Alan. And as a follow-up, so how does this research on, right now it's focused on COVID-19 because of the pandemic and the significance and, just the research going on around the world, but how does this impact our understanding in general on infectious diseases? Because when we are looking at the host genome response, no matter from which population, how do you, what are your viewpoints here? In general, the effect of this research on infectious diseases is the question.
Is that directed to us or to anybody in particular?
Yes. So, no, that's, yeah, as a follow-up to you.
Yeah, again, so I'm sorry that Janet Chow, our immunology collaborator, isn't here. I think Eric and some of the others, and Alex maybe can comment after me, a little more informed. But in general, what we're finding is, as everybody knows, there's a variety of different aspects of the immune system. There's the humoral response and the cellular response, and then interferon response. And we're finding that, as with other infectious diseases, there is normal variation in these. Clearly, the interferon response pathway appears to be implicated. And I think as we learn more about the response to SARS-CoV-2, we are finding that some of the same genes and proteins are involved in response to other immune diseases. So I think as a generalization, what we find here is going to, by and large, be applicable, I would guess, maybe most specifically to other coronavirus infections, but also more generally to other viral infections in general. But I'll let some of the other experts on the panel address this.
Ravi?
I think I agree with Alan. This is more of an immunology heavy question. I'm going to let the other people on the panel take that. I think Alex is looking at it. Go ahead, Alex.
Yeah, I mean, I'm happy to comment on that. I think what we certainly learned from this situation, and I think infectious diseases in general are, in that sense, exciting model disease, where we see that rare, medium rare, and extremely common genetic factors all may play a role in your predisposition, how you fight off any kind of pathogen. And I think that's also the excitement of this consortium, that we bring together the expertise in vertebrate species and evolution, people that have a strong background in common diseases and in rare diseases. And I mean, that's, at least from my very personal background, I'm so fascinated in the rare diseases. So the most severe and end of the spectrum, almost Mendelian diseases and true monogenic factors, those all may be extremely rare, but they teach us so much about the disease. And we have to realize, and I think the NIH just summarized that in a review of the Human Genome Project, that even up to today, we know that we have 20,000 genes in the human genome, but only for 6, 7, or 8,000 or so, we know what they really do. So they're still a long way ahead. And as we see, more and more diseases are raised, and from those we can learn by studying those diseases what the natural function of the respective genes are. So I think that is a fascination on its own right already. And for the rest, I think Alan already laid it out very nicely.
Thank you, Alex, Ravi, Alan. I see Eric is on. Dr. Jarvis, can we do a test? Hello. And Mike, are you on now? Let's see if we can hear you.
No.
Yeah, I think we can hear you, Mike.
You can?
Yes, we can hear you. We can't see you, but we can hear you.
Oh, you can't see me?
No, but we can hear you.
Okay, my camera says that it's working, and I'm showing video on my screen.
Now we can see you.
But if you can hear me, all right. I guess I know that Ravi had asked the question a while back, and you know we have this discussion now about more general infectious disease. Where would you like me to start from?
Wherever. Let's start with the general, and then go to Ravi's question on the criteria.
So I guess, you know, following up on the discussion going on right now, I think that this is, I mean, obviously this is a very unfortunate situation, but it's also a kind of unique situation in the genomic era in that we're going to have a very large number of people definitively exposed to a particular infectious agent that we know nobody had been exposed to before. And I think it's going to be important over the next several years to follow up with people and see what the after effects of that are. And I think there are potentially a lot of interesting studies beyond just the immunology and the immediate response that one can do in terms of what are the things that get triggered by exposure to an infectious agent downstream of that. And we're already starting to see people who are, long COVID cases, people who have neurological symptoms that are not directly tied to the primary course of infection, but seem to be triggered by the disease. And I think it's going to take us a little bit of time to figure out, are these really statistically significantly associated with COVID infection? But for the things that are, I think it's a great opportunity. Unfortunately, our system has been perturbed and we can learn something from that. I think to go back to Robbie's question, I think that he raised a really good point in his introductory statement about the fact that, because this is new, clinical care for both reasons of improvement but also reasons of resource has changed over the course of the disease. And so when we look at the criteria, our goal is to identify people who are true outliers, because we know that there are, patient conditions that predispose them. We know there are environmental conditions that predispose people. And from the perspective of doing rare variation genetics, those individuals are not likely to teach us very much. They may tell us something about common genetics, but when we think about rare variants of large effect, we're likely to see those in the people who are the extreme outliers at either end. And so we've picked a set of criteria that attempts to enrich for those individuals at a level that gives us power to find events that are causal. But it's true that over the standard of care on ventilation, the standard of care on various other drug treatments has improved over the course of the disease. And so, for example, a person who might have been ventilated in April might not be ventilated now. They might be given an alternate course of treatment. And so I think the criteria for what we consider a severe case that we'd want to look at is going to practically change over the course of the pandemic.
Thank you so much for these answers. Dr. Hasty, I think, shall we revisit another
thank
question?
you. Silvio, do you have a connection now? Could you just respond quickly?
But I don't know, can you hear me?
It's very low, but if you speak loud, I think it'll be okay.
Okay, so I'll try to. Okay. I'll try to speak.
I'll re-ask the question, okay? The question was, could you discuss how GWAS can or cannot be used to find and analyze structural variations? And is there a history of GWAS being used to find structural variations?
So how can we use it? So first, we can use it in two ways. First, sometimes we can impute very rare variants, regardless how they are, based on large reference panels, and those rare variants can then be followed by doing.
Silvio, I don't think anybody can hear you. So Alex, if you see in the team chat, do you mind? I think he has also written the response. Do you mind reading that out? Because I think, thank you so much, and we apologize for this audio connection. Alex, go ahead.
Thank you, Silvio. I'll read your answer. So the answer that Silvio wrote is answering the question from GWAS helping finding SNVs, SVs. The argument is twofold. We can impute now pretty rare variants that, if shown to be associated with a trait, can be followed with Bionano. Second, common associated variants and rare variants seem to occur in the same gene. Consequently, common variants associated with regions are likely to support associations with rare variants.
Thank you, Alex. Our next question, Sven, is for Dr. Jarvis. And he has, yeah, okay, all right, Sven, go ahead.
No, he doesn't have any audio connection, so I'll ask him the question and I'll bring it up if he's able to respond by text. So let's just move on with that.
Okay. Our next question is for Dr. Hoyshan. Let's start with the audience question first. So the first question is, do you think there is potential to inhibit STK26? And your observations and your comment from our observations from the study.
Yeah, certainly. I think we don't fully understand yet what this means, but I mean, it certainly is intriguing that we see very high expression in this individual for STK26, that is the gene, but maybe the protein MST4 is even more well known. And that is a negative regulator of the TLR pathways. So with a more active form, that may suppress your TLR signaling, which in response could lead to lower interferon levels again. So I think if we could get back to cells of this individual, then we would be able to test in vitro whether there is an appropriate or non-appropriate interferon response. And that could be some functional evidence that this structure variant in particular may have an effect and add to the predisposition to severe COVID.
Thank you. And as a follow-up, can you just talk a little bit about the role of SVs in immunity and infectious diseases in general, and in our host genome structural variant consortium, structural variants at a population frequency of 20% of less, what in your opinion was used? So what in your opinion would be the most relevant frequency range for infectious diseases? I know that's a kind of like a very broad question.
I mean, yeah, I'll try to. I'll give you my few cents on that. So obviously my background is really in rare diseases and Mendelian genetics. However, especially for infectious diseases, we also still have to consider that someone, while carrying a genetic variant, still needs to be exposed to a certain pathogen, and in this case, COVID-19. So there may be people that carry variants but don't get exposed, hence don't get sick. So that is a threshold that we usually don't see in fully penetrant, otherwise Mendelian disorders. So that is something we should keep in mind. However, to really enhance the chances to find something that has a severely predisposing effect, I still think it's very interesting to start from the really unique rare structure variant side of things. If we then subsequently want to move forward to also finding more mildly predisposing factor, then certainly we can lower the threshold, and that's basically what we do in this consortium, going towards the host genome from either angle, from extremely rare and from relatively common.
Thank you. Thank you so much, Alex. Sven?
Yeah, we have an audience question for Mike Zodi. Have there been any studies looking at COVID combined with sickle cell anemia that you know of? We've gotten this question actually a few times, so there's someone very eager to find out.
I'm not aware of any studies that have been done specifically looking at sickle cell, and without knowing the distribution of the genotypes in some of the large studies like CHGI as far as the ancestry of individuals, I would think that since that should be the sickle cell mutation should be picked up on a SNP panel, that I would think a relatively small GWAS would pick that included a sufficient number of African ancestry individuals would pick up an association with sickle cell fairly strongly if it was present. So I'm not aware that that's been reported. I don't know if anybody else has any other knowledge of people having specifically looked at that.
Ravi? Ravi, we can't hear you.
I'm not, can you hear me now?
We can hear you now. Yeah.
Thank you.
Yeah.
So Sven, we have a collaborator on campus we have a major sickle cell center, and they are looking at COVID-19 and sickle cell, especially in the community locally here. I'm peripherally involved in it. Mostly we're looking at coagulopathies and stuff like that. But I know Medical College of Wisconsin have a pretty big blood bank. I think they are leading the efforts. And if the audience member is more interested, I'll be happy to connect them to that group. And they have their own small group, which is pretty active in sickle cell and COVID-19.
Thank you, Ravi.
Okay, thank you.
Anybody?
Yeah, we had a question for Eric Jarvis that we asked him over chat and that he responded to. So the question was, Dr. Jarvis mentioned that he wanted to use comparative genomics and compare the genome of species that are resistant to COVID-19 infection with those that can be infected. There's been some cases reported in the media about animals in zoos, for example. So we asked him whether there is a precedent for this and if this has been shown before, if this approach has been successful. And Dr. Jarvis replied with talking about the precedent in the original SARS infection and other SARS viruses. So, for example, no bird has been found to be infected with the original SARS virus. And some have argued that this is due to the fact that there are differences in the ACE2 receptor, which we know also plays a role in SARS-CoV-2 that causes COVID-19. And he also mentioned that some species do take up the virus, but do not become ill from it. So something else is happening as well besides differences in the ACE2 receptor. But so overall, this approach to compare animal species that can or cannot get infected and find out what the genomic differences are is really a valid and I think very original approach to this disease.
Thank you, Sven. And I don't know what happened. Welcome everyone to day 5 of our next generation. William. I don't know what happened over there. My apologies again. Okay, we are back to our panel. Alex? We can't hear you. William, can you unmute Alex because we cannot hear him?
Hello, can you hear me?
Yes, now.
Yeah, thank you. So the next question is for Dr. Coley. Dr. Coley, do you see a utility for structural variation findings to be used clinically?
Yes, I mean, that's a very generalized question, but we do use structural variants in clinically, both as a biomarker and as a target. We do have a lot of experience in oncology, and I think when we're coming back to COVID-19, there is so much work still needs to be done, but as a subclass of biomarkers, I mean, we're looking at multiple things from the host genome. It's not just going to be one thing, and as Alex said, we will start with one structural variant in one group, and what downstream biomarker in that particular pathway might be easily adaptable to design and develop. And I think that's how things will progress. Let's say we identified one structural variant A. That variant has downstream biomarkers or pathway which can be easily targeted by an ELISA or a blood prick or a simple PCR. And I think that's the approach most of us will take. We are very early in this, but I'm by the end of what we propose to do is sequence 1,000 genomes on the optical mapping. And I think after that, we should have enough confidence about what structural variant really are involved in susceptibility or making it more aggressive. And then either directly identifying those structural variants sometimes might be challenging, but identifying downstream biomarkers in the pathway as a biomarker. And on the other side of those structural variants is targeting these structural variants. I think some of the trials have already started in few structural variants, and as we keep on identifying more, confirming them in the lab as well as bigger data sets, people will be interested in targeting those structural variants as a treatment approach. Thanks, Alex.
Yeah, thank you, Ravi. I think Sven has the next question.
Yes, it's a question about some of the data in the paper. The paper shows a number of common variants that were picked up with Saphyr, some of which that are more common in African Americans. And Dr. Coley, earlier today, you started by showing that African Americans are also more likely to be severely ill. So I thought to maybe ask the panel, let's just maybe start with Michael and then work our way down. If you can speculate about this, if there's any possibilities that some of these common variants that you identified to be more common in African Americans also may predispose them to this more severe infection. Maybe Michael, you want to go first? Do you have any thoughts on this?
We cannot hear you. Maybe William, can you?
Can you hear me now?
Yes.
Okay. I think it's certainly a reasonable hypothesis that some of these variants might play a role in greater susceptibility based on ancestry. I think we have to be a little bit cautious about that because we also know that socioeconomic status correlates with ancestry and that has a lot to do with exposure and prior history that may predispose people to both infection and more severe cases. And so I think that what we would need is to be able to look at a large number of individuals adjusted for those factors and see if the variants by themselves play a role. I think the advantage we have with the common variants is that there is the possibility that we could impute those into much larger data sets for which we haven't used the more expensive assay techniques to get an estimate of their frequency in larger populations with phenotypes and be able to determine whether those are actually associating in the way one would expect if it's a common variant of significant impact.
Yeah, that makes sense. Thank you. Anyone else who would like to comment on that?
Alan, Catherine?
I was going to make a related comment a moment ago, just in terms of thinking about the relationship of structural variants to some of the other genetic variation that we're measuring. And as Michael is doing, I think it's important that we keep in mind that we want to take a holistic view of the genetic variation that may be responsible here. And so, for example, some of the structural variants that we're finding that may be heterozygous in a particular individual, there may actually be compound heterozygosity that is at play here. And so we need to correlate the short read sequences as well. We need to correlate expression levels that may be influenced by either common or possibly even rare variants and enhancer elements and promoter regions and so on. So that we don't want to be taking any one data type in isolation, but view it holistically together with other ways of looking both at the genome and also at the possible effects of unseen changes on gene expression.
I'd add that it's a blessing and a curse with Bionano because we have such sensitivity of data here that it shows the importance of having a consortium and an adequate number of controls, that we're not identifying things and misattributing them to being causative when it is variation as a result of ethnicity or family background. And it just hadn't been identified before because Bionano is so much more sensitive and gets these regions that other techniques miss.
Yeah, a point actually that we've raised in our consortium discussions before is trying to tease out the relative influence of environment, socioeconomic status, activity levels, and so on, relative to genetic. And so comparison with control groups is critical here. With respect to short reads things, obviously there are large databases like Nomad that have some degree of ethnic information associated with them. But really here we're also like exposure to the dose of virus exposure could have an impact on severity and so on. And so one thought might have been that to look at spousal controls for adults, it's obviously not going to work for children, but they're going to be in the exact same home environment at least, so you're controlling for some factors there. This is going to be a challenge, I think, for all of us, and in particular with respect to the pediatric patients. How do we identify the appropriate control populations?
We have an audience question here that just came in from a person whose mother was moved to the ICU this morning because of COVID-19. And the person asks, if we assume that this virus will persist as a pandemic for years to come, do you think that identification of these SVs should be considered as a future tool for screening patients as a preventative measure to identify those that may be the most vulnerable?
We'll start with Ravi and we'll just move around.
Sure, I think that is definitely one of my personal goals, as well as I know a lot of people working on. It's just not structural variant. It's a whole variety of things we're looking at. Is there something which we can identify a panel or a set of biomarkers at the time of admission and make sure that appropriate care or preventive measures are done at the beginning based on genomic makeup to make sure we have appropriate resources allotted to those individuals as early as possible. And I think this is that direction. So SVs are going to be one of the piece of that puzzle, along with a whole bunch of other biomarkers, depending on the stage that individual is, pre-infection, post-infection, and so on. So yes, I mean, bottom line, yes, we definitely want to include something like this as a biomarker panel for not just screening, but even monitoring at the time of admission, ER, or even in the post-admission.
Yeah, I would
thank
say
you, Dr.
The utility in terms of like deciding whether or not these have impact somebody's risk is going to really be decided by how much of an impact any one factor has. So for the very rare genetic diseases that Alex's group and our group here at the Manton Center study, where if you have a genetic change, then you definitely have a particular disease or condition, that's obviously very important to know and it impacts somebody's medical care. If we're finding out things that maybe, confer a 10% increased risk or a very small percentage increased risk, then there's really not that much value because you need to have a lot of different factors coming together in order for these multifactorial influences to have an effect. So we're hoping, obviously, that what we discover will influence management and prevention, but it remains to be seen, I would say.
Alex Hoyshan, in the case of these severe immune defects that you discovered, did that at all affect anything about preventative measures?
Yeah, so at least for one of the two families from our paper, we had contact to the extended family because the mother was carrier and she got infected, but she was able to fight the virus quite efficiently, as you would expect for an X-linked trait. But then we looked further into the family whether there's any other, particularly male TLR7 mutation carriers, and luckily that was not the case. But my colleagues at Immunology are preparing a patient management plan. So if we would identify now asymptomatic TLR7 mutation carriers with mutations that would lead to a similarly severe deficiency, we would first of all certainly recommend the respective carriers to act extremely carefully to not get infected, but once infected to really be very fast by alarming an intensive care unit and potentially then get treated, for example, with interferons. So in that sense, there may be direct implications, but I think the level of evidence for which genes and for which mutations that may apply, that is still something that we hopefully learn much faster now.
Yep. Anyone else on the team? Mike Zodi, do you have any thoughts on this?
So I think there are a couple levels to this. I think at this point, a lot of what we're working on here is trying to understand the biology of this infection that's really sort of hypothesis generating for treatment. That we're a good distance away from knowing you have a gene mutation, here's a drug. And that for a lot of these things, they may be rare enough that we're never going to get an appropriate clinical test to develop a targeted therapy for people with a certain mutation. From a preventative point of view, I think there might be some value, but looking ahead 5 years when we might have a larger fraction of the population that has prior knowledge about their genetics, right now the time frame to generate these data are not consistent with the time frame for infectious disease treatment in a critical care setting. But from a prevention perspective, we're going to tell you to do the same things that we would tell you to do if you weren't at high risk. There's not a specific risk modulation that you could take because you have a specific mutation. And so I look at this as very much a basic science. How do we understand this disease that can eventually turn into hypotheses for broader therapeutic pathways?
Yeah, thank you. Eric Jarvis, rumors are that you made it on live.
Can you hear me?
We've tackled 10 questions or so. Can you just give us your answers to all of them real quick?
Yeah, well, okay. Well, Alex, I heard your answer to one of the questions for me, and you gave it perfectly. So, on the ACE2 receptor and other things that prevent infection or if infected, also prevent severity. I'll just say on this last point, I think, the point that I was making in my presentation, it seems like a lot of genetic associations are converging on the interferon pathway. And I think that gives hope that if there is going to be some drug in the future that can target something that, that somebody has a genetic susceptibility, targeting something at the convergence of that interferon pathway, I think would be the way to go. Of course, there's more that needs to be done to verify that, but I think that's a good working hypothesis for the community to think about more and to move that possible therapeutic approach, earlier rather than later, focusing on that pathway.
Thank you.
Thank you so much, Eric. And we've had an amazing, panel, and thank you all for coming. And thank you all for your presentations. Ravi, do you want to wrap up the panel discussion? And then I'll take it on from there.
Sure. First of all, thank you guys for making time and presenting what we're doing here. This is just the beginning because we're already looking at host genome and the response to vaccines and stuff like that. So this is going to be a continuing learning process of the host genome, and not just the infection, susceptibility, severity, but the next stage is going to be vaccine response and so on. So thank you, Alka and the team Bionano for putting this together. But thank you all for putting your thoughts together and spending some time with us. So thank you, Alka.
Thank you, Ravi. Thank you, everybody. No, don't go for a minute. I do want to let all the audience know that, and all our viewers and our expert panels, that we've had a tremendous 5 days. This is our 5th day. And we will be, having this Next Generation Cytosymposium on an annual basis, where we anticipate and hope that we will see extensions of this work, additional work, and, be contributing in the field of next generation cytogenomics and, COVID-19 development in research and its applications, for helping patients that are infected. With that, I still need to thank all the people behind the scenes, William and other people, marketing people, and other people that put all of this together, recording of the presentations, and especially to all of the speakers that gave the time and put your thoughts together and, did these recordings and were part of this panel discussion. Thank you, everyone, and we'll see you next time. Thank you.
Thanks, Alka.
Bye.
Thank you. Bye-bye.
Thank you, everyone.
All right, so we have 17 likes to go, and then I'll start my live trading and Q&A session for you guys. So if we can run it up to 150, we'll get started. $15 likes to go, and we'll start. Need 150, and I'll start right now. $12 to go. All right, so we have 141. We need 9 more, and I'll start. 8 more. Come on, run them up. Bye, Idris. Hope you have a good day, man. I want to thank you all for being here. First of all, before we get started, we only have 3 more likes to go. So I want to thank everybody for being here and sharing this knowledge, this information that's coming from all these scientists that are well-renowned. So hopefully you gathered a lot from this today and throughout the whole symposium and my 5 live sessions that I've held on this. And hopefully from this you'll be able to gain more knowledge on if you want to invest, if you don't want to invest. It's really up to you, up to your decision. So, with that said, we have 150 likes, so thank you for that, and we will get started on price analysis. So, volume started to pick up yesterday. That was Thursday's symposium, and we're looking at the 2-day 5-minute chart right now. And currently, I have a trend line set right here. sorry, actually this one right here, and the top of that trend line today ends at around $10.15 or so. So that's a potential upside I see for this stock. If it continues to see the same amount of volume that it's been seeing, and people are super optimistic about it as they have been over the past 2 days, then it's going to continue to go up. But of course, we'll need more volume to support that. There was a lot more volume during this time yesterday than there was today, which is kind of interesting to see. And also, something I'd like to point out is that the Q&A, or actually the whole symposium, was a lot shorter today than it was previously. I think yesterday's symposium wrapped up around 12:45 Eastern Standard. This one was about 15 minutes shorter. And then the one before that was like 3 hours or something like that. But anyway, so let's look at the price analysis a little deeper and kind of zoom in to see where the volume started to pick up and where this could end up going today. So looking at the 2-day 5-minute chart, again, I want to point out that 10.15 that it could rally towards. I think there is a potential catalyst today. I think that Simon from ArcG is supposed to be having, or he's supposed to be posting a podcast, and that should be available over on their YouTube channel. Somebody correct me if I'm wrong on that, but you should be able to look up Arc on YouTube, Arc Invest on YouTube, and be able to find that podcast sometime today. I don't know if it's going to be posted today, but I think that it is, just from what I heard on a Weeble post. And I'm not sure how reliable that source is, but that is just what I know from right now. Yeah, right now I'm really seeing a bullish flag form, and I'll try to explain a bullish flag just to help you guys out to see what I'm seeing here. So we saw this huge spike right here, and then we saw this downtrend, add trend line, and so there's a bullish flag somewhere around this area or so, and it's coming towards the end of this. So what we could possibly see is a huge rally from this. So once it breaks out of this area, and we'll zoom in. Oh, that's too close, sorry about that. Once it breaks out of this area, it could go towards the upside and rally until this trend line right here, possibly. Of course, there needs to be more volume in order to support it going up, and currently the volume is pretty low in comparison to what it was this morning, and in comparison to what it was yesterday around this time. So we have to keep that in mind as well. I want to check the 1-day 1-minute chart just to get a more accurate price movement. Let's see. So, yeah, it's coming right towards the end of this right here, so we could see a break out from this. Towards the downside, if it does end up going down today, I would imagine that it's going to find a support either here at around 827 or so, or possibly around this area, $7.93, somewhere around there. Okay, so Simon tweeted out that their investment conference will be posted for everyone to listen to. That's good information. Thank you, Psych. Yeah. Okay. So keep track of their YouTube channel, or keep track of his Twitter page. That might you might get a better source of where that's going to be from Simon himself. Yeah, as MKC said, we're guessing that it's probably going to be posted to their YouTube page. A lot of I think that most of their podcasts are held on their YouTube page, but again, correct me if I'm wrong. MACD will cross and go up. So we can actually look at the MACD. The MACD does seem to be turning around. That does look like a bullish sign. We could see this blue exponential moving average line cross this yellow simple line, and that would create a buy signal and possibly send it up out of this bullish flag. Currently, we're getting a break out of this bullish flag, but again, there's just not enough volume to just send it skyrocketing like it did this morning, basically. I'll pin that link so you guys have access to their YouTube channel, and that's going to be ARK Invest YouTube channel. All right. So if there's any questions you have about this stock, not the science behind it, because I'm really not that well versed in the science behind it, but if you have any questions about the stock itself, SEC filings, really anything related to that, let me know in the chat. So bullish flag, that it means the stock will rise and stay at a new niveau from there and won't fall again. That's not necessarily, it doesn't necessarily mean that it can't come back down from that, but generally whenever people see these sort of patterns or trend lines, they'll take action on them, usually, is what we see, because a lot of people are watching the stock right now with all the volume that's on it. So whenever more and more people see these different signs, you'll usually get more price movement accordingly. Price target for Monday. Well, Monday is Martin Luther King Day. Market's gonna be closed on Monday, but Tuesday, I honestly couldn't tell you the price for Tuesday. If we zoom out maybe to the 20-day 1-hour chart, we might be able to see my price target. Possibly. So if it continues along this top trend line, we could possibly see it somewhere around $14. But that's only if the volume continues to be high as it has been over the past 2 days and people continue to be bullish on the stock. But that's really going to depend on the catalysts that are coming out over the weekend. I know that somebody in the chat, I don't remember who exactly it was, they said that there's a potential bull catalyst that Bionano Genomics is going to be posting about sometime over the weekend or on Tuesday. But I'll have to verify that for you guys. Besides hype, will make the stock to go to 20. Besides hype, $20? I think that really you need to see, in my opinion, you need to see ARKG buying in. I think that is the ultimate form of validation, because ARKG seemingly is all knowledgeable about the genomic space. Of course, they could always be wrong. You have the hot hand fallacy. If you guys watch The Big Short, where a lot of people think that if a basketball player keeps making shot after shot after shot, more people are willing to bet that they're going to make that next shot. So the same thing could be applied to ARKG, but again, these people are very knowledgeable in their research and a lot more detailed. What's a bull catalyst? Bull catalyst is just any kind of positive news that comes out about the stock. So that would be something like FDA approval, which I haven't seen anything on yet, or that might be something like ARK buying in, but again, that's not confirmed. And that's something you have to keep in mind, that a lot of this stock and its valuation, I believe, is based on speculation of those two catalysts. As a beginner, I ask what is normal in the weekend and when everything starts again on Monday. So usually from Friday to Monday or just over the weekend, you're going to see a huge gap in the share price. And this could be a few reasons, especially when it comes to a holiday, because some countries don't celebrate the holidays that we do here in America. So you might have the foreign exchange market buying into a stock, and that could end up sending the price up. That's something I think we've seen with Tesla quite a few times. But generally over the weekend, you'll see a huge gap up or a huge gap down. And typically over the week, the stock or the overall index, whatever you're watching, is going to fill that gap that it created. And I hope that helps. No, nothing is a stupid question, Mocha. Don't worry about that. All right, does anybody have any more questions? Let's look at the price movement one more time while I wait for these questions to flood in. So if we look at the 2-day 5-minute chart, since that's where they saw the most volume over the past week, we did see a breakout out of this bullish flag, and now it's heading up. And if we zoom in to see this closer, we'll get a better view of what's going on. So basically, you saw this, it broke out of this trend line, now it's soaring up. It could come back down to test this support line now at around $8.50. And then it might make its way back up and rally again to that top trend line that I set. But obviously there is going to be some resistance somewhere around this level at around 889, so you also have to keep that in mind. And pretty much anytime you see like a peak in volume such as this or this is going to create a support or resistance line. So this one would be somewhere around 892 again, somewhere around there, and it looks like it just got rejected by that, and currently it's heading down. But of course it could always break past that resistance line of 892, and I'll draw that on the chart just to give you guys a better view of what's going on. And let me edit the properties and make this pretty, just to differentiate it from all the other lines that I've set. So we saw it come up. Sorry about that. Something just fell off my wall. Anyway. So 894 is our resistance, and then this is going to be our support somewhere around $8.50. How do you know how many people ask for a buy? You can find it depends what brokerage you're using, but you're gonna have to use what is called level 2 data in order to see how many buys there are and how many sells there are at different prices. It's called level 2 data. So look that up along with whatever brokerage you use. Look up brokerage and level 2 data on Google, and you should be able to find that, Mocha. Will BNGO reach the price close to PacBio? What's PacBio at? The comparison between BNGO and PacBio in terms of valuation, it's really hard to determine. You have to keep in mind that they have separate technologies, and while BNGO has better genome mapping technology. PacBio also has different technologies in the genomic space, so you have to take that into consideration. And while BNGO might have better technology, they only have a single technology as of right now. Of course, they could be using all the profits that they're making and all of the they could be using all the profits that they're making and all the money that they got from their shares that they sold to maybe invest into a new technology. So we might see more coming from this company in the future. But again, this is just speculative, and you have to keep that in mind. A lot of the bull catalysts on the stock currently, I think, are speculative. When will BNGO get FDA approval? That's really hard to tell, Hit Beauty. And the reason for that is just because FDA approval, it really depends on the technology itself and how much better that technology is versus already existing technology. So FDA approval varies from technology to technology and company to company. But overall, from what I've seen, FDA approval is a very time-consuming and very costly. It's very costly to do to get that approved. If you make an account on Webull, you'll get 3 months of free level 2. I wonder if there's any brokerages that have just flat out free level 2 data. That'd be interesting to see. Also, Hit Beauty, I just want to bring up something that Simon Barnett, one of the more professionals in this field, brought up on his Twitter page on FDA. And we'll pull up his Twitter thread that he posted about FDA approval, just to give you his thoughts on it. He might have a better understanding than I do as someone who doesn't really understand the process as much. And I will link that to you in the chat. Alright, and I'm also going to pin that just so you guys have access to it. Is E-Trade a good site to manage stocks with? I haven't used E-Trade myself. I do know other traders that use it, like, what's his name? Josh from, totally blanking right now. Josh from the trading fraternity uses E-Trade, and he likes it a lot. Personally, I like to use Thinkorswim for price analysis and kind of the technicals behind price movement, and then I like to use Robinhood because it has the best user interface whenever it comes to buying and selling different stocks, different options, whatever I want to buy. So I use 2 different brokerages in order to try to get the job done, I guess. Maybe redundant, but it's just that the interface whenever it comes to Thinkorswim is kind of hard to deal with. Yeah, you might see, what you might see as a potential bull catalyst after all the symposium is wrapped up is potentially a lot of news outlets trying to cover their thoughts and ideas of what they gathered from the symposium and all throughout all different days of the symposium, and they might have more focus towards COVID-19 since it is the most relevant. All right. Does anybody have any more questions for me? Heard Biden is going to pour a lot of money into this sector and focus on COVID. That might be true. I haven't specifically heard that, but I think that somebody mentioned that on my Discord. So potentially he could be pouring a lot of money into genomics. I do know that he's going to pour a lot of money into renewable energy. So I do think that we're going to see a pretty good boom throughout the genomic sector, throughout the EV sector. And the third one, I think, is going to be data whenever it comes to security and whenever it comes to cloud storage. So I think that's where the next three big waves are going to be in terms of growth in the stock market. I've covered one short squeeze. I think that was on NEO around their earnings report, in that there was a ton of shorts on the stock right before it. I haven't really checked up on that to see how the shorts have changed, but we could talk about short squeezes in this video if you would like. I think that from what I've seen, at least from BNGO, and I'll pull up the data for you guys, I think that the short volume has been decreasing quite significantly, so I don't think that a short squeeze is really in the works here. But we'll pull that up to verify. And if you want to find this out for yourself, you can just type in on Google BNGO short volume, or short interest is the technical term, and that's how many current shares there are short on the company. So current short volume for BNGO is 83 million. I think it was actually I think it was a lot less the other day. So that's interesting to see. So I need a subscription in order to see this. I don't remember that before, but okay. Maybe we can pull up NASDAQ.com. So this will show you all the short interest and how it's changed. So we had a ton of shorts flood in right here between 12:15 and 12:31, and that does seem to be pretty high in comparison to all the rest. But we're gonna have to wait until, I think the data should actually be released today, and we should be able to find out what the short interest is right now. And if we see a ton of shorts flooding into the stock, possibly we could see a short squeeze. But you also have to take into account the days to cover. Currently, or last we know, it was at 1, and the higher the days to cover, the better the chance of having a short squeeze is. Thank you, Dominic. Thank you so much. Real MVP. Thank you. What a baller. Just like MKC said. I think Dominic was the one that gave me 20 for the first or second live stream I held. So thank you, Dominic. Shout out to Dominic D. Yeah, of course, Pretoria. We're just trying to provide the best kind of information that we can on both sides of the coin. That way you can make the best investment for yourself. So MKC is very knowledgeable in this field. She's a lot more knowledgeable than I am, of course. I'm just sticking to stocks, and she knows pretty much, or she doesn't know. She knows a lot about the science behind all of this. So give it to her, because she knows, pretty much, she has a pretty good understanding of what's going on in these symposiums. What are short squeezes? So whenever a short squeeze happens, you have a ton of shorts, okay? So a lot of people are shorting the stock, and this is through borrowing shares from their brokerage. So people are borrowing their shares from their brokerage in order to profit on the stock going down. So they borrow the shares, and then they buy it back at a lower price. But whenever you see the stock continuing to go up, up, and up, these same shorts are usually buying back their positions to cut their losses. And then you see more buying pressure on the stock since there's a lot more demand for it because of those shorts and less supply. So whenever you have more demand, less supply, the price, just like any commodity, is going to go up. Yeah, what Dominic said, basically. You never want to get margin called. All right. Does anybody have any questions left? I think I'm about to go eat lunch in about 8 minutes, but I'll leave the floor open for questions that you might have. We can look at the price charts in the meantime. So currently it's coming towards the support that I set that's at $8.50, and if we break through this, we could see it maybe heading down to around maybe 837 to 828. So there's more volume here, so this is more likely to act as a support than this right here would. Hey, Tony. Thank you, Tony, so much. I really appreciate that. Yeah, of course. I appreciate everybody being here. And all throughout this live symposiums that I've been holding, I really appreciate all the support you've given me on this channel. And, well, I hope that it continues, and I'm very grateful for every one of you guys. Thank you, Asmir. Have a good day, man. All right, doesn't look like there's any other questions, so I think I'm gonna be signing off here. If you have any last minute questions, just let me know right now. Keep pumping those biceps. Too funny, man. Please explain margin call. So what a margin call is somebody is taking out margin or debt in order to buy into a company. So maybe you have like $1,000, you want to take out another $1,000 in order to put more down on the trade. So you're taking out 50% margin on your position, and then whenever you see the stock start to go up, up, and up if you're shorting it, or go down if you're long on it, then the value of what you own, thank you, TQ. Thank you so much. Anyway, so what you own is going to be worth less as the value of your position decreases. And at some point, the value of that is going to come to the point where you need to put more money into the market or into your position. And if you don't do that, then your brokerage is gonna literally call you up and be like, yo, you need to put more money in here, you know? Because like if you're in debt to them and you can't really pay them off, they're not gonna be very happy about that. It's just like a credit card company. If you go without paying them for 30 days, 90 days, whatever, they're gonna be knocking at your door. They're gonna be calling you up and be like, yo, I need my money. Yeah, definitely be careful, Armin. The thing with shorting a stock on so much volume is you could see a ton of more upside and potentially lose money, because there's a ton of hype, there's a ton of speculation going on about this stock right now, and you kind of have to keep that in mind whenever you're playing either long or short on this. Any thoughts on Plug or FCEL? I heard some news, I think it was about Plug. I think that they're building out their infrastructure. I'll have to look more into that, and I will let you know, James. If you head on over to my Discord, which is going to be linked in the description below, I'll let you know my thoughts on Plug and FCEL in more detail. Yeah, Monday the market will be closed. It's going to be Martin Luther King Day, and it's going to be open on Tuesday. It's going to be the first day of the week for the US markets, at least. It might be different depending on which country you live, but American market, the New York Stock Exchange, will be closed come Monday. Yeah, FCEL is a hydrogen play. Exactly, Dominic. Thanks for clarifying that. I'll link my Discord channel in the chat. Just give me 1 second while I pull that up for you. Alright, and I'm going to pin that comment just in case there's a flood of comments that come after it. That way you guys have access to my Discord channel, and that's where I'm going to be posting pretty much every trade that I make. I post over there, and it's completely free. I also do have a premium Discord, which is going to be through Patreon, where I offer these sort of technical analysis breakdowns, and also like live trading sessions like I'm offering right now. And those are on a weekly basis, and I hold Q&A sessions and a ton of different stuff. So if you guys want more of this, be sure to sign up for the premium membership over on my Patreon. Am I bearish on BNGO? No, I'm not bearish on BNGO, I'm just skeptical. Right now, with all of the potential bull catalysts, I'm very skeptical on if they're actually going to go through. So you have the potential of ARC-G buying, which they haven't yet. You have the potential of FDA approval, which hasn't been approved yet. And that's where I think a lot of the valuation is coming from, on top of what we already know about their technology. And now that the symposium is wrapped up and the scientists have shown off what they found, I'm not sure exactly when that next bull catalyst is going to be, but I think that somebody said there's supposed to be some sort of post, probably a recap of their symposium, and that could be another bull catalyst for their stock come on Tuesday. Thank you for being here, Georgie. All right. So I'm off to eat lunch. Thank you all for being here. And as always,